That's quite clear. My name is Victor Rutten. As you can see, I'm from the Utrecht University in the Netherlands. I'm from the Faculty of Veterinary Medicine. I gained a lot of experience in mycobacterial disease and in immunology of mycobacterial diseases in veterinary species. But last 8 years I've been involved in the LRI, so I'm quite well into leprosy now as well. So, but this session, it has only 3 projects to be discussed, so we can afford to give each of you a little bit more time. So don't worry too much about a minute or two, not too much more, of course. And all these reports are towards the end of the stage of conducting the projects. So that means that there's a lot of and rather complete information. So that's, I think, is quite nice, and I hope that you will have lots of questions later on then as well. So I'd like to give word to the first speaker, that is Madhusmita Das. She's going to talk about the identification of leprosy-associated M.Leprae transcriptomic and human host immune signatures that aid in the early signals for determination of type I and II reactions. Madhusmita, please go ahead. Yeah.
Thank you so much. I'll share my screen now. Thank you once again. Good morning, good afternoon, and good evening to all of you. Today, I'll be presenting on to identify leprosy-associated Mycobacterium leprae transcriptomic and human host immune signatures that aid as early signals for determination of type 1 and type 2 reactions in leprosy. To just give a background on what I'm going to talk about is, we all know that reactions occur in approximately 40% of leprosy cases and are immune exacerbations that cause nerve damage and permanent deformities. And prediction of these reactional states using appropriate biomarkers would enable early treatment interventions to prevent nerve function impairment. The current data, which I'm going to present now, consists of the whole transcriptomic expression profiles of M.Leprae and the human host immune markers that differentiate leprosy cases in type 1 and type 2 reactions with those without reaction cases. We had 3 objectives in the study. The first one was to determine transcriptomic signatures, that is variation in gene expression profiles across the transcriptome of M.Leprae that would differentiate type 1 and type 2 from non-reactional cases. The second objective was to examine the circulatory and mRNA expression levels of various genes involved in anti-inflammatory, innate immune system, and antimicrobial pathways that differentiate the type 1 and type 2 from NR reactions. And the third objective was to validate the above identified signatures in a longitudinal design and identify the potential as predictive biomarkers for type 1 and type 2 reactions. This is an overview of what we have done. In the cross-sectional part of the study, total RNA was extracted from lesional skin of type 1 reaction, type 2 reaction, and non-reactional cases, which were then subjected to whole transcriptome microarray for M.Leprae. RT-square profiler array and qPCR for human genes were performed. Bioplex suspension arrays were for interleukins, cytokines, and chemokines were also studied across the study group. And to begin with the transcriptomic analysis, we looked at, from the raw data, we looked at sample clustering analysis, normalization, and fold changes were determined, followed by hierarchical clustering, analysis of gene and expression. Further, the functional term gene expression analysis for gene ontology and notation was done. And these gene expression signatures for the reactional state in leprosy were determined using all the above tests. In this slide, we looked at the sample variability using principal component analysis and found that the variance was noted due to study groups, that is type 1, type 2, and NR cases, and not any other factors. This is a Venn diagram which represents the number of overexpressed and underexpressed genes. Here for each gene, we created a fold change from the prostate signal data, and based on the variance and expression patterns from the signal intensity, the cutoff value was set, which was 0.6. Anything over 0.6 were overexpressed genes, and anything below that was underexpressed genes. In this analysis, we avoided all pseudo genes and focused only on the coding genes. This is a heat map representing all the upregulated and downregulated genes in T1R and T2R. We did an unsupervised hierarchical clustering and looked at overrepresented gene ontology terms. The x-axis here represents the study groups and y-axis the geoannotation term. This is just we found that most of these overexpressed genes in both T1R and T2R were part of integral and intrinsic membrane protein, and the same heat map has been further simplified in these two images, where you can see that we have the number of genes present in each of these assemblies. These are the differentially expressed genes among the antigen coding genes in T1R and T2R, which were the most prominent ones from the study. And we saw a consistent upregulation of M.Leprae specific integral protein coding gene, that is ML238, across the samples. And we did a by prediction of linear B cell epitopes for ML238 as well. We also did R-disper profiler arrays, where we saw that in T1R, mostly CXC chemokines, that is CXCL9, CXCL10, CXCL2, CXCL11, and CD40 ligands, and interleukin-7 IL-17A was overexpressed. And in T2R, CXC chemokines, CXCL10, CXCL11, 9, 2, and CD40 ligand again were overexpressed. We initially planned on doing DCRT MLPA, which is dual converting phosphorus multiplex ligation dependent probe amplification, but due to the amount of RNA present in the samples, we could only do the multiplex real-time PCR. This was done in LUMC, our collaborative partners, and we have analyzed 11 genes, keeping GAPDH as the endogenous control. These are the data from all the 11 genes, the expression and patterns of each of these genes in the non-reactional T1R and T2R. And we found that there was a significant expression of CXCL10 and CTLA4, which were noted in reactional states T1R and T2R when compared to the non-reactional cases. We also did a bioplex suspension array, where we measured the multiplex cytokines, chemokines, and growth factor analysis. And we've done it using 20 leprosy patients, 20 serum samples of non-reactional cases, 20 T1R and 20 T2R samples. Unstimulated and stimulated cell culture supernatant were used. This is a canonical discriminant functions analysis, which was done by our partners. And we've noted a consistent high levels of CXCL10, IL-17A, and IF and gamma in the stimulated T cell suspensions and serum samples in cases with reactional states. In the longitudinal arm, we've conducted UCP FLA lateral flow assays tests. 960 strips were produced in Netherlands and shipped to Karigiri, where 62 samples were tested longitudinally. Dried strips were then sent at room temperature to LUMC for analysis by benchtop UCP reader. From 15 patients, 3 time points, and from 47 patients, 2 time points were collected during the MDT treatment. We initially planned on collecting 100 samples, which each 3 points of time point, which is before reactions, after reactions, and before and after the reactions. But we were not able to get that many cases, and due to COVID restrictions as well, the number of cases in the hospital went down drastically. So we could only collect 62 samples, and upconverting phosphorus lateral flow assays were performed in 6 biomarkers, that is CRP, IL-1RA, IL-6, IP10, S100A12, and anti-PGL1 specific IgM antibodies. The consistent high levels of CXCL10, IL-17, from the cross-sectional analysis, when our partners did a cross-sectional analysis, they were stratified in different forms. One was stratification by the type of leprosy, where LL, BL, and BB patients compared to BT and TT patients showed significantly higher CRP and anti-PGL1 antibody levels. When they were stratified according to their bacteriological index, CRP and anti-PGL1 antibodies were positively higher in the positive bacteriological index cases. And stratification by reaction status at this time points, the patient suffered from a reaction. Higher CRP levels were observed compared to patients without reactions. Further dividing the reactional patients based on the type of reactions, regardless of treatment, it showed that there was increased levels of anti-PGL and CRP, and they were associated with patients experiencing type 2 reactions in this cohort. P-value was determined by Wilcoxon test bed, and the yellow highlights actually are the significant values which were seen. IP10 and anti-PGL1 and IgM were shown to be drastically improved in the RR, NR, and RNR cases. These are the salient findings which we have got from the study. From the transcriptomic data investigated, the top 3 highly expressed genes were ML0247, 2388, and ML1182 for T1R, and ML2203, ML2388, and ML1663 for T2R. And we could also see a very distinguishing marker, which is ML2664, although it was not overly expressed in each of these study groups, but they were completely different in type 1 and type 2R. So from the host immune gene expression profiles, again, we noted that CXCL9 to CD40 ligand IL-17A and CXCL11 were upregulated in T1R were compared to NR, and in T2R, CXCL10, 11, 9, CXCL2, CD40 ligand were upregulated. With biplex suspension arrays, we noted again higher levels of CXCL10, IL-17A, IF and gamma in the stimulated T cell suspensions with whole M.Leprae, and anti-PGL1 antibodies were also estimated in the samples. The conclusion: a gene set signature involving ML2388, ML2664, CXCL10 could be a potential gene expression marker. Similarly, serum levels of CXCL10, IF and gamma, and anti-PGL1 antibodies can be used as serological markers to correlate with reactional states in the leprosy. Thank you very much. I would like to acknowledge all our investigators here, our workers who have done the maximum of the work in Karigiri Hospital, and also our co-applicants, Dr. Gelyuk's team. Special thanks to Dr. Anuj as well, who has helped with the analysis and a lot of testings also. And at this time, I also thank Dr. Sandeep, who has been a part of the study from the beginning and has helped in the analysis. Thank you very much.
Okay. Thank you, Madhusmita, for your nice presentation. We have one question in the chat from Edesa. And the question is how the early signal were operationalized. Did you follow a code until they developed the reaction with serial sampling? And the last one, are the overexpressed genes are causes or outcome of the reaction?
Yes, that is how we have planned the study, and that's why we had 3 time points where the patient would initially be in non-reactional state when he comes to when he or she is diagnosed for the first time, and in the second time point when he has received the MDT diagnosis, and if he has got a reaction case, and then after the reaction. So these were the 3 time points which we are looking at, so that we can do the analysis in all these time points and see whether the genes are overexpressed or underexpressed, which could actually stratify them in 3 different levels. And unfortunately, we could not get 3 time points data for a lot of patients. We could only get 15 patients, so we could analyze only in that 15 patients for us. Yeah.
Did you use your approach also for let's say a household context? I mean, these are of course extending your let's say patient or potential patient kind of reservoir, isn't it?
Yes, and actually we have now started to do the longitudinal analysis. We have finished with the UCP-FLA, but now we are looking at the gene expression marker also from the prospective cohorts. Now we'll be collecting samples from the patients who are in reactions, who comes, because sometimes the patient actually, when they come for the diagnosis, are already in reactions. So it's a little difficult for us to stratify when we like to see them without reactions, with reactions, and then again without no reactions. So we have started collecting the samples based on that, and using this set of signatures, we would like to establish that as well. Yeah.
And that includes household contacts as well, not to what you're getting.
Yes,
now. Yeah,
yes.
okay.
Our target is household contacts. One major difficulty which we face during collecting the samples and especially testing them on household contacts is the ethical issues which rises up, because most of the household contacts do not agree to give their blood or smears. So it's little difficult for us to convey the message and get the samples to actually test this signatures, but we are working on it and we have started collecting few samples on this.
Okay, thanks.
We have a couple of questions in the chat. Anemieke asks, this is for leprosy versus leprosy reaction, not for leprosy versus non-leprosy.
Yes. Dr. Anemieke is our co-applicant, so she's just, I think, trying to comment on that. Yes, that's correct.
Okay, and Diana Lovegood asks, what clinical definition did you use for the two types of reaction?
The clinical definitions used for these two types of reactions were based on the WHO classifications and the lesional appearance and the immunoexacerbations. And we did not, the entire diagnosis was done by our clinicians when they came for the treatment. And based on that, we had separated them as T1R and T2R. We used both clinical and histopathological diagnosis for separating them in T1R and T2Rs.
Okay, if anyone has more questions for Madhusmita.
Maybe I can, please. Yeah. So you were doing this survey the way you did it now. Did you already decide for, let's say, new panels of parameters for potential future use? So
yes,
has your idea about that changed?
Yes. No. We are still following it up with the signatures what we have. The only constraint was the number of samples which we initially promised in the study. We could not meet it, but we will be continuing with the ongoing enrollment of the patients as and when they come to the hospital. This will be now used for the prospective analysis. Yeah.
Yes, Victor, do we have time for one more question?
I think Dr. Irwin has asked me a question. Yeah, I would like to answer that. Yes, so we have actually that was our main goal. So in the cross-sectional part of the study, we took 20 leprosy patients with no reactions, 20 with type 1 reaction and 20 with type 2 reactions. And we have done the transcriptomic analysis on that. And based on those, we have received the gene signatures, which I have just presented.
Okay, thank you, Lucretia. Thank you also, Madhusmita, for this very nice talk. I would like to continue to the next speaker, which is Jessica Fairley from Emory University and a lot of other co-works, which we'll no doubt introduce. The talk will be about use of plasma metabolomics. So that's an additional thing to what was covered in the prior talk. Plasma metabolomics to increase our understanding of metabolic changes in leprosy and associated reactions. Of course, that's with the purpose of doing diagnostics in the end, of course. But the initial study is a proof of principle study, so to see what anything can happen at the end of that. So, Jessica, please go ahead.
Yes, so thanks for having me today. And the title here is essentially the same, but just kind of highlighting some of our findings in both lipid and nutritional metabolism. And of course, yes, like you said, I have many co-authors and many collaborators across Brazil and Atlanta, including those in Belo Horizonte, like Jose Ferreira and Sandra Leon, and others in Governador Valadares, Minas Gerais, Brazil. So I'm going to go ahead. So because I've presented this work before, I'm going to go quickly in the, through the background and methods and focus on the more refinement of the results than what I presented last year. So as many of you know, high resolution metabolomics are a rather relatively new method to studying disease using liquid or gas chromatography combined with high resolution mass spectrometry. There have been a few studies with leprosy in the past, with the notable findings at the bottom there, particularly increase in pro-inflammatory icosanoids like leukotriene and increased fatty acid metabolism noted. This is a adaptation of a nice figure from Macedo et al. That's a new review looking at the association between metabolism and immunity in leprosy. So I'm just gonna kind of look at this and I think at the base of finding biomarkers, we're looking at really what signals what's the marker or metabolic signature of clinical presentation, whether it's subclinical or multibacillary, paucibacillary. So some of the big things that are related to metabolism that affect disease presentation and immune response are one thing, our lipid metabolism that's been definitely characterized in leprosy with the accumulation of lipid droplets in multibacillary disease especially. When lipid droplets accumulate, this stimulates prostaglandin or metabolism of fatty acids into prostaglandins, which can increase regulator T cells and decrease Th1 and lead to immune tolerance. Also related to metabolism are tryptophan, which the indoleamine 2,3 dioxygenase can be stimulated by M.Leprae, leading to decreased tryptophan, increased kynureninins, and Tregs down here. There's also interplay of vitamin D and retinol as well, which we'll look at. So our objectives were to pilot high-resolution metabolomics to identify unique metabolic signatures related to leprosy, immunity, and associated disease states, and hopefully lead to paths to biomarker discovery, increase our understanding of nutrient metabolism. This was our method. So we were finished with enrollment at the end of 2019. Where we are, you can see our study sites at Hospital Eduardo Jimenez in Belo Horizonte and Credeps and Governador Valadares, where we enrolled newly diagnosed patients, and then endemic controls were recruited both from this clinic as well as actually mainly from this clinic, both contacts and non-contacts who were recruited from the dermatology clinic without significant comorbidity. And then the questionnaire, height, weight, and blood collection were performed and samples shipped to the clinical biomarker lab under Dean Jones here at Emory. And then my colleague, Dr. Jeffrey Collins, did the analysis after with some of the platforms that we use, including MummyChug. The metabolites were quantified, compared between groups, and all analyses were controlled for age and sex. This is just our results in terms of the table 1. So you can see we had 42 cases and 25 controls. Clinical forms range from indeterminate to lepromatous, and about 62% were multibacillary versus 38% paucibacillary. There was a few reactions diagnosed at the time of leprosy diagnosis, and then most of the multibacillary had a positive bacillary index, but not all. One participant was excluded from the analyses because it was not a new diagnosis of leprosy, but was a new diagnosis of type 1 reaction, but that individual was not included in the following results. So here's a nice heat map to look at just the overall differences in metabolites. Actually, just let me go back. Just as a note, 13,869 metabolites were detected and describe MS in retention time. So on the left, this is an example of both a heat map and a PCA plot where you can see overall similarities between cases and controls. These are just looking at positively charged metabolites. So on the left, you can see the blue are cases and the yellow is control. So there's about only 2 controls in this kind of section, the similarities on the left. On the right, you can see where most of the controls are. There are some scattered cases where I think no one would be surprised by this, that given the heterogeneity of cases, that they would be some that look metabolically like controls. And you can see over here on the right, also there definitely seems to be kind of an overall separation between cases and controls, with only a couple of the controls over here on the right side. So this is an output using the MummyChug software that identifies pathways that are significantly different. So you can see any pathway that crosses here, the dotted yellow line, are significant. So all of these are significant. This is looking at all cases of leprosy versus the controls. And you can see what some of the common metabolic pathways like prostaglandins, arachidonic acid, linoleate, so fatty acid, some TCA cycle and carnitine shuttle, looking at energy metabolism. If we look at just multibacillary versus paucibacillary cases, some of the same similar pathways are there, like arachidonic acid, but we also see vitamin D, vitamin A, some steroid hormone synthesis, and bile acid. So to kind of see if we divided the cases along positive bacillary index versus negative bacillary index, instead of the clinical MB versus PB, we did that and found obviously some differences because there were different participants in different pools now. However, we saw very similar metabolic pathways listed here, like again, fatty acid, vitamin D3, bile acid, and steroid hormone synthesis, and the carnitine shuttle. So here I'm just going to highlight some of these differences. When we look at the actual pathways, we can then break down the different metabolites in each pathway and compare concentrations across mean concentrations across groups. So here you can see, looking at arachidonic acid metabolism with prostaglandin E2 and leukotriene, which these two work. Leukotriene actually works as a regulator, and there's a balance between the pro and anti-inflammatory properties. So you can see that both paucibacillary and multibacillary are quite a bit lower than controls, but there are some differences among PB and MB as well. These are some more eicosanoids, like metabolites of arachidonic acid. Again, the paucibacillary seems like the lowest. Then we have some different levels between the controls and MB. Energy metabolism was interesting. There's the citric acid cycle and carnitine metabolites were elevated in paucibacillary. And we know that mitochondria are key players in innate immune response, involved in both kind of T cell differentiation and immune signaling pathways, as well as activation. Activation of the inflammasome, xenophagy, and apoptosis. So here you can see that paucibacillary has kind of significantly increased levels of some of these carnitine shuttle and TCA metabolites. Vitamin D, interesting. Definitely been work looking at vitamin D levels in many infectious diseases, and we know that vitamin D has is a significant player in the immune response. Here we see that cases seem to be lower in vitamin D with on the left, and then on the right, Possibacillary seems to have a higher levels of some of the metabolites than multibacillary. Retinol as well, we just see some kind of overview of differences with retinol. On the right, all these are MB versus PB. I included a BI negative, BI positive because these were significant for looking at retinal levels. MB versus PB was similar, but not quite statistically significant. But you can see that retinal levels are seem to be definitely lower in the bacillary positive cases. Looking at some tryptophan, these we found, although we did not find the actual metabolic signatures different, we did find some levels of, oops, sorry. We found some levels that were different among tryptophan between multibacillary and possibacillary, and chimeric acid, which is a metabolite of tryptophan when comparing BI versus BI negative. And we do see some differences here. Looking at the kind of show increased tryptophan metabolism in multibacillary disease. So, so what do we take from all of this? I think the key differences that we found here were related to inflammation and immunity, including the arachidonic acid, which supports a lot of the in vivo and other metabolomic studies. We know that lipid mediators downregulate the important cell-mediated responses as well as Th17. Tryptophan, there's been a lot more interest in this, also in TB, that decreased tryptophan supports catabolism into kynurenines, which again, downstream affect the CD4 T cell responses and can lead to immune tolerance. Some interesting thing look at both with the indole pathway as well as some of the bile acids that there suggests a possible microbiome or gut-derived differences that could shed light on susceptibility to disease. And also this energy metabolism, looking at the importance of mitochondria. This is a new area of study in intracellular pathogens like M.Leprae, but we know that mitochondria maintain cellular hemostasis, and possibly the differences between MB and MB in the carnitine shuttle support this. And then again, vitamin D and retinol differences support looking at implications of nutrition's role in leprosy. So I think a lot of these lead to supporting these in vivo, so finding support some of the recent in vitro literature on tryptophan fatty acid, and open some doors to further avenues of study, looking at role of nutrition and metabolic pathways involved in immune response, which did feature prominently. Taking some of this pilot work to better look at different groups like pre or subclinical exposed, could look at patterns of endogenous or exogenous biomarkers. Our limitations were small numbers that could limit discovery of significant pathways. It was cross-sectional. We had hopes to do more follow-up for reactional states, but the pandemic and some of the logistical challenges limited that. However, we are planning further studies in a large NIH-funded project where we will be recruiting 1,200 community participants in a hyperendemic area of Minas Gerais, identifying anti-LID1 positive and negative. We'll be following these participants over time to look for development of clinical disease, and we'll be doing metabolomics at several points along the way. So hopefully can take some of this preliminary work to help do some targeted metabolomics of these pathways to help look for kind of metabolic signatures and biomarkers. So happy to answer any questions. Of course, acknowledge the large team involved in participant recruitment at both FASE and Universidade Federal de Juiz de Fora. My colleagues at Emory, colleagues at Fiocruz in Rio for helping with some of our logistical coordination. So happy to answer any questions.
Thank you very much, Jessica. It's I think good to see that you were able to finalize the project anyway, in spite of the pandemic that you suffered from. And I heard you say in your last slide that you're going to aim for targeted metabolomics. That's the idea in the end, isn't it? So, but this is proof of principle, so you show that there are differences. So I was wondering.
Exactly.
If TB would be there in the background, which usually is, isn't it? How could you differentiate?
Yeah, that's an important consideration. We definitely, I mean, we asked questions about, medical history for sure, and we did not have any people that reported TB, but obviously people could have latent MTB, which is hard to differentiate, with some of our markers. So that's something something important to consider how we can approach that with further studies. Yeah.
Okay, so I think that Untitled is doing metabolomics of TB patients as well, I guess. Well, at least in our surroundings, so it might be nice to compare these and see whether there's comparable things, because in the end the mycobacteria are both intracellular, so which may cause similar issues. Thank you very much. Lucretia, please take your chances.
Thank you, Jessica, for your nice presentation. The first question is from Anemieke, and she asks, what mechanisms do the metabolic pathways that are discriminatory for leprosy refer to, and what can you say about the mechanism WRT leprosy pathogen?
So I guess if I'm understanding, I think the biggest question that this brings up are these pathways we're seeing a result of the clinical case. So are we seeing MB versus PB, the downstream effects because of the subsequent, immune, already differentiated immune response, or are we seeing some signals that might have predated this time and led to some of these differences? So I think that's obviously some of the big question. If I'm, but I may not be really addressing Anemiek's question. I don't know if you want to follow up, can do that.
Anu asked other question, and is were the patient that did not cluster together with the patient group on the right side of the heat map similar in the leprosy classification or bacterial index?
That's a really good question. Actually, that we went through and did that because I was interested to find out what these cases were, and they were actually all the way across the spectrum. So I was interested to see that maybe these were the indeterminate or the paucibacillary, but really we found it across the spectrum. We individually went and looked at every case that was over on the side of the controls, and we're all over. I think one thing, we kept consistently seeing a lot, if you saw, paucibacillary kind of differentiating itself from both the controls and MB. Obviously the controls included contacts, and were we just seeing a stimulation, a difference because of a robust immune response in paucibacillary, or were we seeing some similarities between MB and the controls because of some other reason? Every control was examined thoroughly for any occult signs of leprosy. So, but I think that our bigger study, where we'll be able to try to catch some subclinical disease, will be interesting to compare to these results.
Got two more questions. I think we can deal with these. Lucretia, just go ahead, but we must take it a little bit short in time.
Had asked, are these different in metabolic specific for leprosy or comparable to difference in other infectious disease with various levels of bacterial load?
Yeah, that's a good question. A lot of these were similar with TB. Our lab at Emory is really before we did this, and why we went down this road is because of their experience with TB. And my colleague Jeff Collins, who does a lot of the analysis, he has some interesting findings, very similar with tryptophan and other. So TB is the biggest one I have to compare to, but I think you're right that a lot of these do, the intracellular pathways could be very similar to other intracellular pathogens. I think the fatty acids though is somewhat more particular to leprosy and possibly in TB as well, other mycobacterial.
And the last question is from Richard Truman. With the low broad distribution of results seen, especially on multivariate cases, do you suspect that more definite classification of cases using really European scale or similar refinement might benefit the analysis?
Yeah, that's a great.
Is it possible to do that?
Yeah, that's a great question. I mean, we didn't end up getting biopsies on everyone, although there were clinically kind of diagnosed based on the Ridley-Jopling. Unfortunately, with the low numbers in this, we couldn't. I did want to do the lepromatous versus tuberculoid or something like that. And unfortunately, the numbers kind of when you go down on the numbers, obviously the significance kind of goes away, and that's a little harder to deal with. But again, yeah, that's something that would be better to do, and that's why we, when we shifted to BI versus BI negative, we did have, several of the MB cases who were, BI negative go over to the other side, which at least the differences from the large view were very similar, the metabolic pathways, which was reassuring that even in possibly, we were still seeing that MB, even with a clinical diagnosis, was differentiating itself.
Okay, thank you, Jessica. I think we
Sorry.
must stop here. Thank you, Lucretia, as well. I would like to give word now to the group of Pushpendra Singh, and it's Anuj Mavlankar who's going to give the next presentation, and that's about experimental infection of armadillos. Just go ahead, Anuj.
Good afternoon, all of you. I am Anuj Mavlankar, and I am going to present today on the project entitled Biomarkers for Early Detection of Leprosy using Comparative Transcriptomics. So to give a brief background and introduction to the project. As we all know that laboratory diagnosis of leprosy is challenging due to complex factors like host genetics and the long incubation period of the disease. So these factors make human transcriptomic studies in leprosy quite difficult, owing to these factors like uniformity and the duration of exposure or the infection in an individual. So to curb this, the 9-banded armadillo is the only animal model for the leprosy, and some of the armadillos are also able to resist the experimental inoculation of Mycobacterium leprae. So in this study, we have utilized the armadillo as an animal model for identifying the specific biomarkers of the leprosy progression by means of comparison the susceptible and the resistant animals using RNA sequencing. So susceptible animals are those in which the disease has been progressed after the inoculation of M.Leprae antigens, while the resistant ones do not show such signs. So the objective of this study is to identify the candidate biomarker genes which are indicative of the disease progression in experimentally infected armadillos using RNA sequencing, followed by bioinformatic analysis. And the relevance of this study would be to it would help in developing a suitable target for developing point of care test so that it can be utilized for early diagnosis of leprosy. This is the part host transcriptomics experiment, and it has been carried out in National Hansen's Disease Program, that is in USA, and which serves as a basis for this project. So in this experiment, basically 10⁹ bacilli at a specific time point has been injected to the selected armadillos. These armadillos are captured and born. These were captured and raised in captivity, and they were then injected with M.Leprae antigens. And as you can see, post 4-month infection, the PBMCs, that is the peripheral blood mononuclear cells, were extracted out of these armadillos and at specific intervals. So followed at the month of 24th month, the seroconversion, impaired NCV and disease progression has been seen in 4 of the armadillos. And impaired NCV here is the impaired nerve conduction velocity, and seroconversion means the detectable number of antibodies has been seen. And similarly, you can see no seroconversion has been seen in 6 of the armadillos, so they will be considered as a resistant to M.Leprae. This is the overall outline of the work which has been carried out. The host transcriptomics experiment, as I have already explained, which involves the revival of the cryopreserved PBMCs and followed by the M.Leprae stimulation and RNA extraction. Once the RNA is extracted, the RNA-seq library has been prepared and followed by Illumina sequencing. These sequencing reads were then considered for the differential gene expression analysis, followed by the pathway and the network analysis of the candidate genes. Also, we have compared the candidate biomarker genes with the leprosy transcriptomic studies published in databases like the Gene Expression Omnibus, which I'll come to that part later. So in the differential gene expression analysis portion, the cutoff criteria we use to shortlist the candidate biomarkers from PBMCs is p-value of less than 0.05, that is a 5% level of significance, and the log fold change of greater than and equals to 1. So in all, with the total number of expressed genes from armadillo, a total of 331 were found to be differentially expressed, of which 39 were upregulated and 292 were downregulated in susceptible armadillos, that is those showing symptoms, signs of leprosy progression. These genes were then subjected to the gene ontology enrichment analysis, which showed the enriched terms to be neuron differentiation, the projection development, and the muscle structure development, among others, as you can see in this picture as well. This is the volcano plot where the green the genes in the left portion represents the down-regulated genes, while the red ones being upregulated, isolated from the PBMCs, and the right one showing the dot plot, which represent the specific enriched gene ontology term for these set of genes. So mostly they are lying in the leukocyte differentiation or immune response or the defense response, which invoke specific immune cells in leprosy. Further, we carried out the network analysis where the protein-protein interaction has been studied for these 331 differentially expressed genes using the string database. Here you can see the PPI enrichment values, which is quite significant. The hub genes for these network has been computed using the betweenness centrality algorithm of the cytoscape. Here, cytoscape is used as a tool for this particular thing. The hub genes, like as you can see, the green one, ACT-A2, which is the actin, which is actin like smooth muscle alpha-2 actin, and is found to be involved in the notch signaling pathway. Basically, previous studies also report notch 1 and notch 2 being the two important receptors which have an important role in leprosy pathogenesis. Like it interacts with the transmembrane ligand, which is called as the DLL, that is delta-like ligand, and it is a portion of basically the antigen presenting cells, and it has a significant impact during leprosy progression in an individual. So this thing, and IL-6, caveolin, and DCN and MMP2, which is a matrix metalloproteinase 2, it has shown to be an important role in T cell signaling pathway, which I will explain in the next few slides. As you can see, this is the figure A represents the network for these enriched 331 differentially expressed genes, and figure B represents the color of these genes represent the hub genes which have been shortlisted using the particular algorithm. ACT-A2, as you can see in this table, has more number of genes, has more number of degrees that it shows that it has some relevance in that particular network. Similarly, you can see all the other genes as well. Next portion in this study is the comparative analysis, where we have compared the differentially expressed genes with those of the other leprosy transcriptomic studies. As you can see in the image, 2 data sets from Gene Expression Omnibus has been taken, and the number of genes which are differentially expressed in each of the studies has been compared. So, excuse me, it is coming out to be 9, that is, which are common among these 3 data sets, despite of the differences in the experimental conditions of the studies where they have been carried out. So as you can see, IDO1, CXCL10, IL6, these are some of the significant genes, as you can correlate with the previous presentation of Dr. Jessica as well, where she was explaining about the IDO1 tryptophan, IDO1 role, which is helpful in the tryptophan metabolism and showing and lead to immune tolerance in individual. So that could be quite significant. IDO1 has also been reported in the recent study by Calvo et al., where they have compared the leprosy and the non-leprosy skin lesions using RNA sequencing, which indicates the robustness of these genes as the potential biomarkers for developing a point of care test. As you can see in this image, which it is basically a T cell signaling pathway, which has been designed using cell designer software and the markup language, that is systems biology markup language, has been used. So you can see in this pathway, IDO1 is quite significant as it directly relates or have impact on the NFKB gene, which is nuclear factor kappa B, and similarly other genes which we have identified using the network analysis, like caveolin 1, then DCN, which is a decorin protein, then IL-6, interleukin-6, then MMP2. So all these are quite significant as they have a potential role in during leprosy pathogenesis in the T cell signaling. So some of the genes quite activate, enhance their expression levels during the progression of the disease. So these could be considered for further exploration. So these are basically the genes which we have identified in this study, IDO1, which catalyzes the first and the rate limiting step of catabolism of the tryptophan, as also explained by Dr. Jessica. CXCL10, as explained by Dr. Madhusmita as well, that it shows an activation of CXCL10. It plays a vital role in the neuron in response to the brain injury for activating the microglia part. Then IL-6 cytokine, which helps in the inflammation of the maturation of B cells, elevated expression during the bacterial infections, ACT2, MMP2, caveolin, and DCN. So of these, the IDO1, CXCL10, IL-6, and MMP2 have been previously been reported to have a potential role during leprosy progression. So these markers would be considered further for analysis. And the new ones which we have reported being ACT-A2, caveolin, and DCN, which could be explored further. So to conclude this study, the comparative transcriptomics has immense potential to identify candidate biomarkers and to understand the complicated molecular mechanisms associated with the pathogenesis of leprosy. So our findings reveal key candidate genes like IDO, CXCL10, IL-6, ACT-A2, MMP2, caveolin, and DCN. So these could be the potential targets for early diagnosis and must be further utilized in a larger sample of population and patient samples. So it would be helpful. I would like to acknowledge the funding agencies, the LRI, Leprosy Research Initiative, and the Turing Foundation, R2 Stop Canada, NHDP, National Hansen's Disease Program, where the overall host transcriptomics experiment has been carried out. I would also like to acknowledge Dr. Pushpendra Singh, my guide, where the lab, where the bioinformatic analysis and this part has been carried out. ICMR Jabalpur, and all of you for your valuable time and attention. Thank you so much.
Thank you very much, Anuj. I have sorry to tell you that we have just a few minutes, so 1 or 2 minutes to answer questions. So maybe Lucretia, you could to go to the two questions that are there in the question and answer. I was summoned to come back to the main room at 16:22, which is now. But well, I take the risk of going on for 2 more minutes. Okay, go ahead, please.
Thank you, Anuj, for your presentation. We have a couple of questions in the chat. One is from Anemike, and she asked, how do you see the development to point of care for transcriptomic or host markers? How do you see the development to point of care for transcriptomic host markers?
It's basically the low level have been observed at in the resistant armadillos as compared to the susceptible ones.
And Anuj asked where CXCL10 and IL-6 expressed at higher or lower levels in the resistant armadillos compared to the susceptible.
Basically, the CXCL10 and IL-6 expression were checked and they were found to be lower in resistant armadillos as compared to the ones in which the disease has been progressing.
Thank you.
Coming to the second question by Dr. Anemiak.
Sorry.
Coming to the second question by Dr. Anemiak, she asked how this can be actually converted into point of care test. So basically, this is for identification, candidate identification. But so this, in a similar way, as you have done, Dr. Anemiak Galuk, anemic help, that these candidate genes can be used in using some lateral flow assay model. Either it can be potential target for UCP LFA or for other form of lateral flow assays to make them field friendly. I hope this answers to your question, Dr. Anemi.
Okay, thank you.
Do we have time for other questions? Because
I think we need to stop now because we have to go to the plenary sessions. So hopefully, Anuj, you can have a look at the questions and answer them in the chat still. So they are not going to be lost in that case. Okay, well, I have to thank Lucretia for support and moderating this session, and I have to apologize for letting it go that long. So we couldn't finalize the discussion, but we had a nice discussion overall, I think. And then I think, and what you showed this quite nicely is that that there's a lot of correlation between the different approaches. So that's something to work on in the near future. Thank you very much, and I'll ask you to leave now here and to go into the plenary session.
And just 5-second last correction.
Thank you to all presenters.
Live bacilli that was used for infection and not the antigen, that was mistakenly mentioned by Anuj. So that was a small correction. Thank you.